like a catalyst. Okay, so every enzyme will do what? It will speed up the rate of the reaction, but it itself will never participate in the reaction, right? It is just a, how it works is, it's just a facilitator, okay, a helper. So what it does is, it lowers this activation energy. So an enzyme that does not have a reaction, that does not have an enzyme, which is this one, right? It will have much higher activation energy, as you can see. But when we use an enzyme, the red line is the reaction that is occurring with the enzyme, right? So you see how much this energy of activation has lowered. So this is without enzyme, this is with enzyme. So you see it has lowered so much energy, and then that's why the reactions occur much faster when we use the enzyme, because enzymes function by lowering the activation energy, and they do not affect the Gibbs energy, the free energy. Okay, that is completely unaffected by the enzyme. So that's all you need to remember, that the enzymes only act as a catalyst, and how they speed up the reaction is by lowering the energy of activation, which is called EA. Okay, and every enzyme needs to have a specific substrate to join. Like I told you, sucrose can only break down when it gets its specific enzyme, which is called sucrase. Okay, lactose will only break down when it gets a specific enzyme, lactase. Okay, maltose will break down only when it gets its specific enzyme, maltase. Okay, now you would think most of the times you can get the name of the enzyme from their substrate, but then at the same time there are different names also. Like starch can break down only in the presence of enzyme called amylase. Okay, so that's not starchase. Make sense? But most of the time you can see, like I already gave you example, sucrase for sucrose, lactase for lactose. Make sense? But you have different enzymes. Most of the time you can tell their function also from their name. Okay, but here the key point that I'm focusing on is, why I'm saying is that every enzyme needs a specific substrate. If it does not get its specific substrate, it's not going to be an efficient chemical reaction. Okay, so why this happens is, when there is a substrate, you see this is a enzyme. Okay, let's say this is lactase. Okay, so now lactase needs a specific substrate. This is called the active site. This site is the active site. So the active site is on the enzyme. And why do we call it active site? Because this is where the substrate will come and bind to it. Okay, let's say the substrate has to shape like this. Okay, so that it can fit in here in this shape. Make sense? Now if the substrate is shaped differently, let's say its shape is like this, now it cannot fit in this space, right? So physically it should be able to fit in the active space. Now this cannot fit in here, so this will not be the correct substrate for this enzyme. Make sense? So basically the substrate and the enzyme, their shape has to complement with each other. Meaning, just like for every lock, physical lock I'm talking about, there is a specific key, with that only you can open the lock, right? Same way every enzyme has a specific substrate, and it will bind to its active site only then. Okay, so this enzyme has to be shaped like this. Okay, make sense? So this is now complementary. It doesn't matter what the other shape is. Here it can look like this or this, but as long as the shape where it has to go into the active site is complementary, it's going to work. Make sense? So that is one of the things. That's why only a specific substrate is needed, and when the enzyme meets its substrate, so this is its substrate, right? Then it is going to form a enzyme substrate complex. Okay, enzyme substrate complex. Okay, so that enzyme substrate complex now breaks apart. Okay, so the activation energy is going to loosen the bonds, and then lactase will separate into two separate products. One is called glucose and other is called galactose. So because it's a disaccharide, so it is going to form these two molecules. So that's how this whole reaction will occur. So lactose is the substrate. What is the substrate for here? Lactose. Sugar is the substrate. So this substrate will bind to the enzyme at the active site, and enzyme substrate complex is formed, and then it is going to release into products. That's how all these chemical reactions work. Okay, so here they have given example of sucrase, but I gave you a different example of lactase. So you can always read this, and in addition you have this one too. Okay, so active site is what? Is often a pocket or a groove. Groove means what? Like a valley, like mountains and valleys. So that is what I was showing you, that it is like a groove here. In this it will fit. Make sense? And the shape of the substrate is responsible for the specificity of the enzyme. Now, when the substrate enters the active site, the enzyme changes its shape slightly. You know, when you have to insert your key into a physical lock, right? You have to little bit jiggle the key sometimes, right? To get the right fit. So same way here also, enzyme, when the substrate tries to fit on it, the enzyme will change, like little bit jiggle or wiggle, you know, kind of that, to have a tightened grip on the substrate, right? Like a handshake, you know, that's what this says. So this is called induced fit, and this results from the interaction of the various chemical groups of the substrate and the active site. Okay? So this is what we call the enzyme. This is your enzyme, and you see this is the active site. Now this substrate will fit in here only when there is a exact match of this space to this space. Make sense? So this, now it really fit into it. You see that? It went in here. This is called enzyme substrate complex. And now this will break down into products once the correct substrate bound to the correct enzyme. Make sense? So substrate usually is not bound. When it sits on the active site, it is not a very strong chemical bond. It's usually a weak bond, which can, so just by little jiggling, wiggling, it will break off and form the products. Make sense? It is not a very strong bond. But it happens, the reaction happens very fast, and it very quickly it will break down into the products. Okay? So this is one other active site and the enzyme showing. So these are, see, these are the two substrates for the sucrase enzyme. So they are going to fit. You see how there is a little notch here? So this substrate which has a notch only will sit on this active site which has a notch here. You see that? Only then. Now this substrate has a little notch or uphill over here. This only can sit in this active site. They cannot go other way around. Make sense? That is what we mean by that lock and key method, you know. So now this have fit very nicely in the enzyme. Now they are going to they have found the enzyme substrate complex. Now this active site will lower the enzyme will lower the activation of energy, and you see the substrates are now converted into products. They were red and green, and now they have become yellow and blue. So they are the products. Now with little bit action, substrates are going to get converted into products, and now these yellow and blue products are released, and you see enzyme is gonna go back unhurt, unaffected. This was the actual shape of the enzyme. It is released after the chemical reaction just the same way it was. So nothing happens to the enzyme. Only it is a facilitator, I told you. It just helps the reaction to run faster, okay, by lowering the activation energy. Okay, so that ends this particular portion, and now we will see the factors that affect the enzymes, which kind of what you did in the lab. So the rate of the enzyme reaction can be sped up. Sped up means you can make another video. You know what I mean? Like you can end